Hi everybody and welcome to Regents Review Session 4. This is Ms. Winans. Today we're going to cover the central dogma of biology, that is how to go from DNA to RNA to protein. During protein synthesis, you're going to have three components of the cell that play a major role. You're going to have the nucleus, which we all know holds the DNA, the instructions to make the proteins. We have the ribosome, which is where we have the center of protein synthesis. And then we have the cytoplasm, which is going to be where we will collect the amino acids needed to make the protein. In the first stages of protein synthesis, we're going to go through the process of what is called transcription. Okay, in the central dogma of biology, we have to start with DNA, we have to transcribe it into RNA, and then we can translate it into a protein. Transcription occurs right here when we go from DNA to RNA. DNA can't leave the nucleus because it's the original copy, whereas RNA can, because if RNA gets damaged, we'll still have the instructions embedded in the DNA protected by the nucleus. So transcription is going to happen in the nucleus. Okay, so we're going to take the template strand of DNA, we're going to make a complementary copy of RNA. We're going to remember to use U instead of T because this is RNA. So in stage 1 of protein synthesis, we enter transcription, and DNA is going to provide the instructions to make proteins. The resulting structure is called mRNA. The M stands for messenger. mRNA is the message from the nucleus to the ribosome. It tells the ribosome what protein it's supposed to make. As we enter stage 4 of protein synthesis, we are in the process of translation. Okay, remember the central dogma again is going from DNA to RNA. That RNA is called mRNA. It's the message from the DNA to the ribosome, and then we're going to make a protein. This process occurs at the ribosome. Okay. So what's going to happen is that our ribosome, which is located right here, will have our mRNA. Our mRNA will have our codons. Every 3 bases on an mRNA sequence codes for a specific amino acid. We're going to introduce another type of RNA, which is called tRNA. tRNA is known as transfer RNA, and it's going to transfer the correct amino acid to the ribosome so that the ribosome can start developing and building the protein that we need. This ensures that we don't just make any protein, we make the exact protein that was coded for in the DNA. To better understand what type of protein you will make based on an mRNA sequence, you can use the universal codon chart. Not even the scientists who study proteins know all of the codons and which amino acids those codons code for. So they too will use the universal codon chart. The way you use the universal codon chart, let's say you had a codon that was UCA. The way you would do this is you would start with the first base, which is U. So I said UCA. Okay. The second base is C. You'd move your fingers together to match the first and the second base in a box. Once you get the first and the second base, you can either use the third base over here, or you can just look for the codon you're looking for. You'll notice that this codon codes for the amino acid serine. Every so often there is a mistake in the DNA. Now, mutations are random and they're rare. We have a really good editing system that involves our DNA that when mistakes occur, usually we can fix them. But every once in a while mistakes do happen and it can result in either a beneficial, neutral, or harmful mutation. The two types of mutations that you need to know are a point mutation and a frame shift mutation. Sickle cell anemia over here is an example of a point mutation. Point mutations occur when one point in the DNA is changed. So in this case, where we're supposed to have thymine in the DNA, we have adenine. So therefore, if we take a look at this, it'll change the codon, 3 letters on an amino acid sequence. So where we're supposed to have the codon GAA, we have the codon GUA, which results in a different amino acid. So what happens here is this structure allows for the tube-shaped red blood cell. Whereas if we take a look at this structure due to the mutation, we get the sickle-shaped red blood cell. In this case, it's not a neutral mutation because the amino acid did change. So this is either going to be a beneficial or a harmful mutation. In the case of sickle cell anemia in the United States, it's a harmful mutation because this sickle shape can increase the chances of blood clots and reduces the individual's ability to transport oxygen. But in areas where malaria is very prevalent, due to the shape of the sickle cell, since malaria is a blood-borne disease transported by mosquitoes, people with sickle cell anemia cannot get malaria. So in areas where malaria is prevalent, sickle cell anemia is actually a beneficial mutation. But in areas where malaria is not present, sickle cell anemia is a harmful mutation. So that's an example of a point mutation. Okay. It also sometimes is referred to as an inversion because a different base is put in place of another base. Okay. Over in the other side, we have what is called a frame shift mutation. So a frame shift mutation is either going to be a deletion, where a base is completely removed, or an insertion. In this case, what happens is the G that is supposed to be in the DNA for some reason gets deleted. We're supposed to have this amino acid sequence in our protein. But if we take a closer look, when we delete that G, it shifts the reading frame. Remember, every 3 bases on a DNA codes for a codon on an mRNA, which codes for 1 amino acid. So with the removal of G, all of these bases in terms of counting our codons shifts, and we end up with ATG, AAG, CAC, GT. That's not a codon because it only has 2 letters. So where we were supposed to have a protein that had 1, 2, 3, 4 amino acids, we now only have 1, 2, 3. So we shifted the reading frame and created an entirely new protein. So again, a point mutation is where one point of the DNA has the wrong base. A frame shift mutation is when a base is either removed or inserted, shifting the reading frame and changing the codons. Cloning. Mother Nature's clone is identical twins. What happens in development? We have a sperm, we have an egg, they come together, they create a zygote. When that zygote splits to form body cells through mitosis, it results in 2 embryos. These are supposed to be circles, they look like squiggly lines. 2 embryos instead of 1. Since they split from 1 zygote, their DNA is identical. So Mother Nature's clone is identical twins. However, scientists have figured out a way to manipulate cloning, and it's shown in this picture of the sheep. So what happens with cloning is we will take a body cell which has a full set of DNA, a diploid set. It has 2 copies of every chromosome. And then you have an egg cell which has half of the genetic information. It's haploid. We talked about this when we talked about mitosis and meiosis. An egg cell will start thinking that it's a body cell, a zygote, once it receives information from the sperm, half of the information from the sperm, receiving a full set of DNA. Scientists have found out that you can trick the egg cell by removing the egg cell nucleus and then inserting the body cell of the organism that you want to clone. So now the egg cell has a full set of DNA. It thinks it was fertilized by a sperm, but it wasn't. What happens is it goes through the normal development phase. We all know that in the normal development phase, we have fertilization, which leads to the zygote, which leads to mitosis, which leads to the embryo. So once we insert the nucleus of sheep A into the egg cell of sheep B, the egg cell thinks it was fertilized, so it goes through mitosis to create body cells to create the embryo. Now we need to insert this embryo into a place for development. So in mammals, the place of development is the uterus. So we find a surrogate. A surrogate does not have to be related to the clone or the individual who donated the egg cell. A surrogate is someone who's just donating their uterus for the development of the baby. So what we do is we implant this into the uterus of the surrogate, and then however the gestation period time for the sheep, it will develop and it will be born, and then we will have Dolly, which was the first cloned sheep done by scientists. So what happens in cloning is that we trick an egg cell by taking a nucleus of a body cell that has a full set of DNA, inserting it into the egg cell after the egg cell's nucleus has been removed. This tricks the egg cell into thinking that it's been fertilized. It creates an embryo. We implant the embryo into a surrogate mother. The mother then gives birth to the clone. The clone will be identical to the individual that donated their nucleus of their body cell. So in this case, Dolly is the clone of Sheep A. Recombinant DNA technology is exactly what it sounds like. It's when we recombine DNA. We use this technique to make insulin as well as human growth hormone. What we need to do is we need to extract the gene that we want another organism to produce. So what we do is we use these enzymes called restriction enzymes. These are enzymes that cut DNA. In class, we referred to them as bioscissors. Please don't put them down as bioscissors on your Regents, however, because that's not an acceptable answer. So we're going to focus with the word restriction enzymes. So what we can do is we can go into the human nucleus and we can extract the gene that we want produced by another organism. So let's say this is the gene for human insulin. We then take a plasmid from a bacteria, which is a circular piece of DNA, and we use bacteria because bacteria can reproduce very quickly within a day. So what we do is we take their plasmid and we also cut the plasmid with our restriction enzymes, and that allows a gap. We confuse now the human insulin with the bacteria to create recombinant DNA. Recombinant DNA is the DNA of one organism combined with the DNA of another organism. We insert that into a healthy bacteria. We allow that bacteria to reproduce. And now we have these bacteria that can not only maintain homeostasis as bacteria, but they can also produce human insulin that can be extracted and distributed to diabetic patients. This is extremely helpful because before this technology, we actually had to use pig insulin for diabetic patients. This caused allergic reactions in some of the patients. Once we were able to use this technology to create human insulin, the allergic reactions were reduced. If you want further assistance in understanding how recombinant DNA technology works, please also know that it's also considered genetic engineering. Please look at this video down here for further assistance. Gel electrophoresis allows us to separate DNA samples using charge according to size. The way we do this is we take different DNA samples, and again, we use our restriction enzymes. Remember, these are enzymes that are able to cut DNA. What we do is we take samples of DNA and we put them into DNA wells. We cut up the DNA using the same restriction enzyme. We insert the DNA into the wells, and we set it up to a charge system. DNA has a negative charge, so it's attracted to the positive charge. So when we set up this system, it's important that we put the negative by the wells and the positive charge down towards the bottom. We turn the charge on, and that is going to pull the bands down the gel. The gel is a matrix, so it kind of has like this net. So the DNA bands have to kind of weave their way through the net. The larger bands are going to get stuck at the top, whereas the smaller bands are going to get stuck at the bottom. Once we turn the charge off, we can then stain our gel and take a look at it. And then we can start comparing the bands. Organisms that have the same bands in common will be most closely related. So we can use gel electrophoresis for determining evolutionary relationships. They can also be used in paternity tests as well as in crime scene investigations.